Hello everyone, I am Varun Aggarwal doing second year Zoology Honours from Hansraj College. Today I'll be talking about Drosophila melanogaster, a very popular and celebrated model organism in biological research. So first I'll be talking about the etymology, that what exactly the word Drosophila melanogaster means. Okay. So if we talk about droso, the droso word means dew or humidity or moisture, and phila means loving. So the Drosophila melanogaster survives on a 60% humidity, hence it is Drosophila. Talking about melanogaster, so melano is related to darkness or black color, and gaster related to gastro, related to abdomen or the belly. So the Drosophila flies have abdomen with have dark banding, dark segments, hence melanogaster. Okay. So now classification, the biological classification. So I have taken this classification from NCBI taxonomy browser. So the super kingdom is Eukaryota, kingdom is Metazoa, phylum Arthropoda, subphylum Hexapoda, class Insecta, subclass Pterygota, infraclass Neoptera, cohort Entopterygota, order Diptera, suborder Brachycera, infraorder Muscomorpha, superfamily Ephydroidea, family Drosophilidae, subfamily Drosophilinae, tribe Drosophilinae, genus Drosophila, and the species which we are talking about is melanogaster. There are approximately 1500 species. One of them which we use in our research majorly is melanogaster species. So the subgenus is Sophomora, species group melanogaster group, and species subgroup is melanogaster subgroup. There are further subgrouped species also. Okay. Now, as we know that Drosophila is a very robust model organism. Why? The question is why it is so. Okay. The one of the very first thing is it has a cosmopolitan distribution. It is found in almost every continent of the world. Different species are found in different continents, as you can see in the map. The Drosophila melanogaster species, which we are talking about, is also found in almost all the continents except Antarctica. Apart from that, it has a very short life cycle. The life cycle goes for around 12 to 15 days with a lifespan of around 2 to 2.5 months. So starting from the very egg, then there is the larval stage. The larva has three substages, which we call as first instar, second star, third instar. Okay. Then we have pupa. It can be also divided into pre-pupa, pupa, and post-pupa. Then the adult will eclode out, and then adult will mate. Oviposition will take, that is the egg laying, and then egg, and this cycle will further go on. Okay. Genomic similarity. One of the very key feature of Drosophila is it has a whopping 60% similarity with humans. We have 96% similarity with chimpanzee as well, but if we are talking about research, I cannot research easily with chimpanzee. Research with chimpanzee is done at the very higher levels, but at the level of basic research, fruit flies seems to be best option. We have 80% similarity with mouse as well, but again the results with mouse take around a year, and with Drosophila we take around 15 days. So that's why we prefer fruit flies. Along with that 60% gene similarity, we have 70% similarity in the genes which causes disease in humans, and 50% of the fly protein sequences have mammalian homologue. This makes our research validatory. That's why we have to do research on this fly easily. It has a low chromosome number, only 4 pairs are there, and these are chromosome 1, 2, 3, and 4, and we can easily study them. It has a high mutation rate, but it's not a bad thing. The mutations are very easily traceable. The mutations can be easily seen in the phenotype. For example, this is a red plus white eye mutant, and this is a white eye mutant. By looking at the Drosophila fly from naked eyes, I can see what changes are there. There are mutations in wing. The wings are curled up in the mutated flies. Like this is a open wing, there are curled wings. Okay, this is one of the most fascinating mutation I have seen in Drosophila's history. It is called an antennapedia mutation. It is caused due to mutation in Hox gene. This structure which you can see is a leg. So in the place of, this is a Drosophila head, in the place of antenna, Drosophila legs were there. This is a normal one in which there are an antenna, but like you can see, legs have grown due to mutation in the Hox gene. Drosophila is very easy to culture. It can be cultured in conical flask, vials, plates, bottles, very easy to culture. In a California fly facility, approximately 60,000 vials are being studied by 30 research groups. So this is the benefit of using Drosophila. It requires very low maintenance. Okay. Now sexual dimorphism is very much evident in Drosophila. I have 4 features earmarked here. One of them is abdominal bending, like this is a male and this is a female. In male you can see the abdominal end has the segments, dark bands fused, but in the female the segments are segregated and not fused. Okay. Along with that, there is a size difference, with the male being shorter in size and the female being longer in size. If we talk about sex combs in male, in the forelimbs of male there is something called sex comb, the spot you can see here, and in female this is absent. Okay. If we talk about abdominal ending, so this is much evident now. This is a male and this is a female. So male has a rounded abdominal ending and the female has a I would say narrow pointed. Okay. Giant polytene chromosomes are present. It is one of the very famous feature of it. This is the salivary gland of Drosophila L3 stage larva, and this is the polytene chromosome which has been obtained by staining. This is an enlarged view of the polytene chromosomes. Okay. Now we have talked about why the Drosophila is such a robust model organism. Now I would like to mention that Robin Mackey, a science editor in the Guardian, says there is a noble love affair between Drosophila and Nobel prizes, not the Alfred Nobel. Okay, so this is because over many Drosophilists have been working tirelessly from hundreds of years in Drosophila. 10 Drosophilistes have been awarded 6 Nobel prizes, which include T.H. Morgan, Muller, and the latest one being in 2017 of Jeffrey Hall. So the first award was given to T.H. Morgan in 1933 for the role of chromosomes in heredity, the chromosomal theory of inheritance. Then in 1946, H.J. Muller was awarded the Nobel Prize for production of mutations by X radiation. In 1995, Lewis, Volhard, Wilches were awarded Nobel Prizes for genetic control of early embryonic development. In 2004, it was awarded to Axel and Buck for odorant receptors and organization of the olfactory system. In 2011, Hoffman, Butler, and Steeman were awarded for activation of innate immunity and dendritic cell and their role in adaptive immunity. The latest one, 2017 only, Hall, Rorschach, and Young, they were awarded for their studies in molecular mechanism of circadian rhythms. Okay. So by this image, I want to convey that the fruit flies are said to be the queen of genetics or Cinderella of genetics. So that implies that if you study genetics, Drosophila has a very great place in genetics to study. But I would like to mention this also, that it's not only the genetics in which we can study Drosophila. Almost all the domains of biological sciences, you say, Drosophila has been studied widely. Let me list them some. Developmental biology, evolutionary biology, toxicology, molecular biology, genetics, neurobiology, cell biology, immunology, cancer biology, neurodegenerative disease like Parkinson, physiology, biochemistry, depression, obesity, reproductive biology, biotechnology, nanobiotechnology, sleep disorder, anything you say are can be easily studied using Drosophila flies, and they are being studied. Okay, now let's delve deeper into the life cycle of Drosophila fly. So this is a comprehensive view from the embryo, which is stationary, then it will hatch out in 24 hours to give first instar larva, then another molting will take to give second instar larva, then another molting will give third instar larva, then the pupation will start to give a white pupa, then there are chitinization and sclerorization to give a brown pupa, I would say a mid pupa, then again metamorphosis will take inside, and this is a later stage of pupa in which you can see the eyes of the adult which will be going to occlude out, and it is the most late stage of pupa in which we can not just see eyes but also the wings, and we can also determine the gender of the adult by the bandings, the early bandings. Then the fly will occlude out like this, and then they'll mate, they'll produce eggs, and the cycle shall go on. This is an image of the copulation of the fruit flies. Okay. Now, each stage at one time. This is an egg. This egg has a chorion layer with dorsal appendages, and sometimes these appendages get twisted, and sometimes rare often these appendages are often are also not there. Okay, so this is a dorsal appendage, chorion layer, nucleus, cytoplasm with yolk cell, vitelline membrane is there. So this is the general structure of egg. Larva. Larvas are important to understand. So there is a size difference first in the first instar, second instar, and third instar, with a gradual increase. Along with that, third instar is a wandering stage. The third, if we culture Drosophila in a bottle, then we can see third instar larva on the walls. It will be climbing on the walls. The second larva almost on the surface of the diet or just above the diet. If we talk about the first larva, it will be inside the diet. That is also a way which we can differentiate between the instar stages. We can see the anterior spiracles in the third instar. The anterior spiracles pop out of the body. The thing which we use in our daily day in labs is posterior spiracle. The first instar larva has posterior spiracle which I would say not visible. The second instar has straightened posterior spiracle with little orangish coloration, and in the third instar it has prominent bright orange colored posterior spiracle which can be easily seen, and this is how we used to identify. So this is the general structure of a larva. It has got anterior spiracle, there is a mouth hook, there is segmentation there, and there are spiracles, posterior ones, and this is a cuticle layer. Now the pupa stage. So this is a early pupa, since you cannot see the eyes and the body of the adult. In this you can see the eyes. Okay, so there is a mid pupa, and this is a late stage of pupa, since we can see the eyes as well as the wings. Okay, this pupa is just about to occlude. Again, the general structure of pupa: anterior spiracle, puparium, the caudal spiracle. In this you can see the layer of the pupa and the body of the adult which is going to be occlude out. In this you can see bristles on the body of the adult, and you can also see the segmentation is like the fly which will occlude out of is going to be a female, and the eyes are also visible. So after the occlusion, this is how it look like. So this is a female. You can see on the segmentation, as soon as it gets occlude out, the flies do not open up, so they are curled as of now, but later stages it will open up. If it continues to be closed, curled, then it's a mutated fly. Okay, now let's talk about adults. This is a image in which adults are mating. First talk about the anatomy. So anatomy goes similar for male and female, except the fact the narrow pointed ending, the dark fused bandings, sex comb, and size different that I told earlier. So there is an antenna, there is a head, there is forelegs, there is halteres, there are bristles on the body which you see in the pupa itself. This is the abdomen, this is the thorax, this is the compound eye which has over 800 ommatidia per compound eye. Okay. So you can see this is a female adult fly. You can also see the iridescence in the wing, and this is the mouth part of It has sponging mouth parts to suck from fruits which are rotting or ripened enough. Okay, so this is a closer view. If we talk about the parts, so this complete mouth part is called proboscis, with this called rostrum, hastellum, and then labellum. Now let's talk about the mating behavior. Drosophila flies has a complex mating behavior in many steps. So the first step is orientation. The male and female shall orient in a particular way to initiate the copulation. The next step will be tapping. The male shall tap the female's genitalia. The male then sing a courtship song with the vibration of its wing to lure the female. It will eventually lick the female's genitalia, and then further it will attempt to copulate. If the female is agreed to copulate, then it will bend its lower part, and if not, she'll kick the partner out. So if she agrees, the copulation take place. So after copulation, this is a pregnant Drosophila fly. As you can see, it has a swollen abdomen. So when if we dissect at this stage, we can see the eggs, the early stages of egg inside her abdomen. This is an image of live egg laying. Like this is a female fly laying the eggs, and we just saw at that particular moment. Okay. So this was all about life cycle. Now, as we mentioned again and again, that is it utilized very much in the research. So we have to conduct some set of standardized experiment which are called bioassays in general. So there are a lot of bioassays actually, but some of them which are very famous are gustatory assay. It is used to see if the diet, if the experimental diet which we are giving is being eaten by the larva or not. So we use dyes in this. There is a climbing assay in which we see locomotory behavior of the fruit flies, that if we have given some drug, are they not moving, are they showing retarded motion, or they are showing accelerated motion. There is a fly weight assay, that we measure the biomass, is there an increase or decrease. We do morphometric, we measure the length of pupa, adult and the larva, if there is any increase or decrease. So these bioassays help us in deciding whatever experiment we have started has some effect on the phenotype or maybe the length of the body, whatsoever the experiment is. So that was all for today, and thank you so much for watching.